Good afternoon. Welcome to the Port Townsend Marine Science Center Future of Oceans lecture series. Yay. I know. This is season 12. As I've said before, we have this wonderful privilege of being able to bring in experts and discoverers and explorers from all around our region who are really on the front lines of looking at what the challenges are facing the marine environment, but also who can give us hope for the way forward. And so every single lecture has been just wonderful this year, and today is no exception. If you would like to get email reminders about the lecture series, there is a little mailing list out in the lobby. Just put your name on there. We won't put your name in a raffle or send you unwanted emails about anything else. So a couple of quick announcements. One is, I'm just going to keep doing this because we started a book club called Dive Into a Book. Right now we're reading Nick Pionson's Spying on Whales, and that discussion is going to be March 8th, and the group meets upstairs from Aldrich's on Sundays, always on Sundays at 2 in the afternoon. Did I say that right? Yes, 2 to 3:30. So if you're a reader and you want a group of nerdy readers, this is the group. And I'm going to make a shameless pitch for our upcoming spring fundraiser, the Tides of March. This year, it has moved back to March, where it belongs, because it's called the Tides of March. And this year, our theme is Making Waves. We're really excited. There's going to be great food, a lot of really wonderful activities, and kind of an exercise part where you get to practice raising your arm with a number on it to make a donation. So if you want to come hang out with us, you should come. It'll be really fun. And March 21st is not that far away, so get your tickets soon. I want to thank, where did the Darrows go? There you are. Thank you to the Darrow family for your ongoing support of this series to make it possible for everyone to come, regardless of ability to pay. We just want you to feel like you're welcome here, and so thank you for doing that. And thank you to everyone in the room for your contributions today. Your donations help support this series, but also all of the other free or very low-cost public programs that we do throughout the year. So thank each and every one of you, just for being here, but also for those little paper bills with the people's faces on them that you stick in that glass thing out there. We appreciate it. I want to thank our volunteers today, Mary, Steve, and Rosemary, for giving up your beautiful Sunday afternoon to be here. Thank you for your help. Thank you to Centrum. Oh yeah, no problem. Thank you to Centrum. Micah and Austin are here today. We really appreciate your expertise and your good-naturedness, and don't know what we'd do without you. And thank you to Washington State Parks, but especially the staff and the rangers here at Fort Worden. They are wonderful partners for us, and we really appreciate them. And also just quick thanks to KPTZ Radio for all of their great programming, especially Nature Now. We sometimes have people interviewed for the series ahead of time, but also Nan and all the interviewers at Nature Now have great programming, and so listen in. All right, same format. Talk first, ask questions later. Chris will get to talk. I will have a microphone and I will bring it around so that he can stay here basking in the light, and I will bring the microphone to you so you can ask questions at the end. Today's speaker is Dr. Christopher Kelley, who some of you have heard before. He was in our series a few years ago to talk about deep sea mining, which was, even at the time, a worrisome thing, and now probably you've heard even more about it in the news under the current administration. So if you would like to go back and hear his talk on deep sea mining from 3 years ago. It is on our YouTube channel, so you can tune in. He also gave a really great talk on the ocean as the Earth's largest biome back during COVID, which is also on YouTube. He's provided wonderful resources for us for mapping data that we've used, and he's just he's one of those people that when you say, what would you like to talk about, his interests and his expertise are vast and varied, and so really just landing on one thing to have him talk about is kind of hard, but I think today, you'll be surprised. So he and his wife moved to Port Townsend in 2019, and probably before they were unpacked, he was already at the Marine Science Center offering to volunteer for us and help us with projects. He also volunteers for Jefferson Land Trust and for Wooden Boat Festival, and I'm probably sure some of you have heard him speak there as well. Before all of that, before we usurped all of his time, he was a program biologist for the Hawaii Undersea Research Laboratory and graduate research faculty for the Oceanography Department at University of Hawaii for 18 years. He's led multiple deepwater research cruises, conducted deepwater fisheries projects, and led a video annotation laboratory that identified deepwater fish and invertebrates recorded using submersible and ROV operations. And the result of all of that work is he has 2 species, deep water animals named in his honor. One is a sea star and one is a coral, which is kind of cool. And that's probably why he went into it in the first place, to get animals named after him. He still serves as onshore scientist for marine research cruises in the Pacific and continues his work, the work that you'll hear about today, on glasswater sponges, and he says in his basement lab, which sounds a little nefarious. But the other thing that he's working on that we could have heard about today is he's working on locating and getting information about a historic shipwreck off Wake Island that he's actually doing for Ocean Exploration Trust, because their ship, the Nautilus, is going to be in that area doing ROV dives later this spring. So that's what I mean. His interests are all over the place, and he has an amazing memory for minutia as well. So if you ask him a question, it's likely that he can dig up the information for you. So ask good questions at the end of today's talk. Please help me in welcoming Dr. Christopher Kelley.
It's actually because I bore easy that I keep switching topics all the time. I'm not sure how to operate this thing, but I'm going to give it a go. So I work on sponges, amongst other types of things and other animals, and this is the very first time I've ever given a talk on glass sponges, and it took a while because to try to figure out, when you've never spoken on the topic before, how to get it to flow right. It's a challenge, so hopefully it'll flow okay today, but excuse me if it doesn't. But I'm going to start off with a real basic question. I'm going to focus mostly on their morphology and their glass skeletons, which is absolutely unique for animals. But I'm going to start out with some basic questions like, why the heck are they calling these things animals? All right, here we go. Okay, so there's a couple reasons. They don't make their own energy like plants. They're not autotrophic, they're heterotrophic. They actually have to acquire their own nutrients, and all sponges do this through filtering, pretty much, except for a certain group of demosponges, which I'm not going to talk about today, that are actually predatory sponges. But anyway, like other animals, they have to get their food from some other source. They have specialized cells, but they also have these cell syncytiums, or tissue, I guess you could call it tissue, that's multinucleate. So it's not necessarily very discrete cells, one nucleus per cell. They can have a lot of nucleus in one massive coating of tissue that covers parts of their bodies as well. So it's different. But they have organelles and they have nuclei, which is similar to animals as well. Of course, that's similar to plants too. And they also supposedly, according to Google AI, because I didn't know this, is that they have neuroid cells that are potentially precursors to neurons, but they don't have a nervous system. They don't have any really developed tissue. And also, they can reproduce sexually or asexually like a number of other invertebrates as well, although that whole process is quite different. And it was problematic for quite a few years where to put sponges, because they do things very different than other animals. And so this is kind of the current status of the animal kingdom. This whole box is the animal kingdom. And in the blue part, which is the slight blue part, all except for the very top part, the peripherans, they're considered eumetazoans, or true animals, because they actually have discrete tissue and they have organs and things like that. And then the periphera used to be known as parametazoans, or beside animals is the definition of that. And they decided, well, that's not very good. So they're just leaving them as periphera, which is the scientific phylogenetic or phylum name for sponges. Poor animals is what it means. So they're off on their own, and they also branched off extremely early in the animal kingdom. They are one of the oldest animals that's still alive. This is extant animals right now. There are fossils that date back to the Cambrian, at least 540 million years ago, and possibly as old as 760 million years. There's a couple fossils that are in dispute, right, whether they're sponges or not, because that's really old, and that would put them into the Precambrian. But one thing why they do have a fossil record for them is they're probably the first animal, if not one of the first animals, to actually develop a skeleton. And by that, most people think of skeletons as what we have. We have an endoskeleton, but skeletons can be an exoskeleton. It basically means they were able to actually create minerals, or I don't know the right way to say this, they were able to mineralize parts of their bodies, to harden them, to give them structure. And that was novel. That was not happening before sponges, apparently. Right now, there are 4 major classes in Porifera, the phylum Porifera, the sponges. I'm only going to talk about the bottom one, the Hexactinellids, because those are the ones that are the deep sea sponges, which most people associate as deep sea sponges. However, there are a few members of the other groups that do get into deep water, and then there's a few of the Hexactinellida that get into shallow water, but for the most part, the Hexactinellids, the glass sponges, are restricted to deep water. And the shallowest one that I've ever seen when I lived in Hawaii was 350 meters down. But when you get up here north in colder temperatures and darker seas and so forth, you can get them in a scuba diving range. So for the rest of this talk, I'm going to pretty much show you sponge morphology, the diversity of their morphology, and then I'll get into their skeletons, because what I want to do is I want you to really just get amazed at how many different varieties there are out there, what they've managed to do over 500 million years of evolution, how many different forms that they've actually developed into, and same thing for their skeletons. And I'm going to start with a video, and it's kind of like it's not the same, but it's similar to a scene that got me very, very interested in sponges back in 2003. And it's a video that actually comes from the ship Okeanos Explorer in 2017 when I was out on a cruise with them off of Johnson Atoll. So let's do this. And I liked this video and said, you're going to have to put up with it. So.
Jeez.
Yeah, you're right.
Talk about a Dr. Seuss-like landscape. Wow.
Look at this. This is amazing. If we could just hold the scene for a moment.
Wow.
This is Attack of the Sponges here.
You wanted sponges, Chris.
Yeah, I got them. Yeah, okay. I got them. Wow.
So the great thing about this that I love is the way they're all oriented directly into the current. This one looks like a Muppet.
This is why sometimes I think these sponges, when you come up against something like this, you think you're on a different planet. Just so strange.
Wow, it goes on and on too.
Every time we do these dives, all I can think about is, this is the type of experience someone would have if they found like life on another planet. Everything's so alien to, at least for me, even though I've been doing it for 5 years, it's still like I'm on another planet when we're down here.
Yeah, and it's definitely the same for me as well, pilot, because even though I work on these sponges, a scene like this is just extraordinary.
And I'll toss my hat in the ring too, as a person who studies echinoderms, which are even more bizarre. These things just stun me. Like a forest of the weird.
Anyway, these aren't just sipping sponges. These things are different than the typical thing that you might see in shallow water. And that's just a sampling of just maybe 3 or 4 of the forms out there, and they just go on. And I'm going to show you some right now as we go through these. But when I started this game back in 2000, when I was hired by the Hawaii Undersea Research Laboratory, I was a fish guy, and I knew a lot about deepwater fishes and things. But then when I got the new job as a program biologist, I had to learn all of the animals. I couldn't just stick to fish, and I was supposed to be able to give a name to any animal that showed up on ROV or submersible video and put it in a database. Well, here's the problem. I don't know what the sponges were. I'm looking at this going, I don't know what the heck this thing is, and not very many people did. So what you have to do is you have to come up with a name for something. You can't just put sponge 1, sponge 2 in your database, because that's meaningless. But if you put something descriptive and you call every time you see that sponge that same name, then when it does get an official name, you learn what it is. You could actually change it in the database, and you're moving ahead. So this is an easy one. I'm going to ask you guys what do you think this thing is. Anyone want to care to answer? E.T., yes, easy. Okay, a little bit harder. What about this one? Okay, this one's skeletal. Okay. How about this one? Okay. I called it the bearded helmet, but... And by the way, these have names. That first one is recently published, same as this one as well, by the Chinese that have been working on some of these sponges. Okay, for those of you my age or older, you ought to be able to get this one here. Close, yeah. Well, maybe that's right. The gramophone sponge, of course, right? All right, this one, kind of easy. The giant leaf sponge. And this one here you probably won't get. But this is our pilot of our submersible dubbed this one. This is Jabba the Hutt. So whenever we see these things, we can, know in the database, and now we do have scientific names, at least the genus for most of these, so it actually helps. Okay, here's the problem, that whenever they do more and more dives with ROVs or submersible, more and more of these sponges we've never seen before crop up, and we don't know what to call them. And this is one in the opening scene. This is in the Mariana Trench, about 5,000 meters down, and we couldn't collect it because we were on a geology mission here, and I could kick myself because we have never, ever seen the sponge before. We can only guess what family it's in. And here's another one. This was down, I think, off of American Samoa. Reminds me of some type of a spaceship or something. I don't know. I can't even come up with a name for the first one. This one, does anyone have? Lasagna, that's true. That's a good one. This one here, I call it the atomic sponge, but, it kind of looks like that. Although that was a sad event, but. This one, I look at this, to me, that looks like the exhaust pipes of an 18-wheeler, if you ask me. And, okay, this one, the purple people eater, I don't know. One of the few glass sponges that has some pigment. And then this one here, I was looking at the Apollo 11 documentary a few weeks ago, and I thought of a name for this one. It kind of looked like the LEM lunar module taken off from the moon, bringing back Armstrong and after their moon walk on here. If you look at that video, you can see the analogy, but anyway, no one would get that one, so you probably skip it. But I don't know what these are. These were not collected, so they await description. The thing is, with the ones you just saw and the ones you're seeing here, all sponges have got to solve the same problems. They all feed kind of the same way. They all are on the bottom. They don't swim away. They got to worry about predation. They got to worry about just sedimentation or how to do things. But they've got a million different ways to do it, which is really bizarre. And so there's a couple structures or body forms I want to go through that maybe we kind of understand it a little bit, but a lot of them we don't. So for example, this one is kind of a sort of a tubular sponge or a vase-shaped sponge. And this is pretty well suited when you're in an environment where the current can come from any direction and switch on you, because the water actually comes into the side of the sponge. Let's see, I think there's, okay, it comes in the side of the sponge and then goes out the top through the main oscutum like that. So it hits side up, side up. So if the current's coming along from one direction or the other, they're round. It doesn't really matter. They've got appropriate surface exposed to the current, and then it's going to go straight up. The other thing about it is that with the currents passing over the opening at the top, you get a Venturi-like effect where it's actually drawing water through the body. It's pulling it like blowing through a Coke bottle or something like that. It pulls the water through. So this is a really good design, and there's a fair number of sponges that have this kind of design. And now there's other sponges that don't have this kind of shape, but they want to increase their surface area, which allows them to actually filter a little bit more. And that one over there, this one right here, this is kind of like a department store where you get into that sponge and you're just looking for socks or something like that, and you go through the whole freaking store. They don't want you to leave their store. They want you meandering around. And it's the same thing here. The water's got to go through all this tortuous route to get out. Increased surface area with all types of folds and holes and all types of things. This is a very common theme with sponges to improve the efficiency of their filtering for food. Let's see. Okay, so that's one of the things. Another thing is that, how many of you have heard of the benthic boundary layer? If that's, some of you, basically there's friction of the water going over the seafloor. Right? And so the closer you are to the seafloor, the slower the water current is until you get up to about a meter, and then that's about all there is in that particular layer. But filtering animals like to get up a little ways away from the seafloor in most cases. And so in some of these, they've got stalks. And they've got all different types of stalks. You've got super little thin ones. You've got divided stalks, thicker ones to support larger sponges here. All different ways, but presumably that's to get up off of the bottom to avoid sedimentation, which is going to clog them a little bit, but also to get into a little bit better current for their feeding. Okay, these guys, I call them the corset sponge because it's like they were wearing a corset. So they're actually creating some kind of a stalk with their bodies rather than actually creating an honest-to-goodness stalk. That's not considered a stalk, by the way. That's just part of their body wall. And same thing here. So that's another way that some sponges have figured out how to get a little further up off of the bottom. And in this guy, I call him a freeloader because this guy, he should be on the bottom, but he attaches very frequently onto these higher dead sponges or even live ones to get up above. And so that's another sort of way in which they figured out how to get out of that benthic boundary layer, but some sponges don't worry about it. Okay. So attaching to the seafloor, you got to be stable in relative to the current. If you're moving along same with the current, you're not filtering the water, right? You've got to stop and let the water pass through you. They've got to attach firmly to the seafloor. They got two ways of doing it. One of them is by gluing, like where they actually add more glass and silica right to the bottom and attach it right to hard rock substrate here. These are all gluers, is what I call them. Like here, and just different forms. Again, it doesn't matter what the form is. These are just a group of sponges. That's the way they like to attach. And then we've got these guys, and I call them grapplers because they've got specialized spicules or specialized glass parts that come down and anchor into the rocks and crevices, and they grab a hold like Velcro. They've got little hooks on the ends of them, and they hook onto there, and it's very, very effective. You could even be hanging upside down or sideways, and it's fine. They're very, very good at this. But also, in my opinion, I think the grapplers are more advanced because I think they've got the best system. And the reason for that is that this system enables sponges to actually inhabit sediment areas, not just the rocky areas, whereas the gluers, you can't glue in sediment, but you can grapple, and these become functioning like roots for them. And the same thing, this is a nodule, a manganese nodule pile here, and they can survive in that type of substrate with pebbles and nodules and everything else because all their little grappling is all around this type of thing, and they're perfectly stable. And then in order to one of those freeloaders, in order to attach onto a dead sponge, that's how they do it. They grapple onto the body, which you can't really glue onto this dead sponge mass, but you can grapple it, and they can get up and survive, and you can get all types of good feeding going on. Okay, another problem, how do they avoid predation? Well, we don't know that, but we do know that there's not that many predators for glass sponges. And there could be a couple reasons that people have suggested. If you take a look at this one right here, this thing is paper thin, okay? This sponge, like most sponges, is 90% glass. There's hardly any tissue worth eating when you eat these things. Now, if you could be a lover of peanut butter, and I take a piece of glass and smear peanut butter all over it and give it to you, are you going to eat that thing? No, even if you love peanut butter. You know, there are probably some snails that can maybe scrape some of the organic matter off of the top of these things, but just being made of glass in itself is a really, really good defense. You know, animals that have calcium carbonate skeletons like corals, they can get eaten. Your digestive system can actually break down calcium carbonate, the acid in it. But what can you do with glass? You can't do anything with glass. These are very, very thin again. And then other types of sponges, they'll put spines on that are a little bit meatier type of sponge, if you will. You got all types of spines where you're going to look very unattractive to eat. This is what's called Walteria here as well, all types of things. Plus, they've got some other organisms in here that makes them a little bit unpalatable. So they've got various ways which they can defend themselves. And this is a hypothetical way. I had to get this example in on this talk because this is something that I've never seen before and never seen since. This was a sponge in a big massive sponge, and we took a sample right out here, and almost immediately, right where we took that sample, it started turning red, right where the hole is. And when we got the specimen up to the lab on the ship, you could clearly see it was red. We don't know what that is, and we have never, ever seen a sponge do that before. Usually you sample them, they stay white, nothing changes, but it's got some compound in there, and the only thing I can think of is that's probably some type of an anti-predator defense, some type of a toxin. A lot of people talk about them. I'm not sure there's a whole lot of proof for toxin. There's a guy who did some work on settling where, a graduate student, where supposedly this particular sponge he was studying had an anesthetic that put larvae to sleep so they wouldn't settle on it. Okay, so there's so much we don't know about these sponges. It's very, very, very likely that they have toxins that are going to kill animals, and animals eventually do not, they leave them alone. Okay, there's another thing too. There are certain species of sponges that have close associations with other animals, usually cnidarians. This is in a genus called Periphragella, and all those little bumps on there are little tubes that I think the sponge makes, and anemones live in those little tubes. And that probably helps defend them, the sponge itself. And you can see the sponge is perfectly white here. It's healthy and it's happy. In fact, when you see them perfectly white like that, they're in very good shape. See, these branches are brown. That's usually dead here. But anyway, that's perfectly healthy, and he doesn't mind all these anemones living in them as well. Same thing for this one, all types of these zoanthids growing all over this branching sponge as well, and it's fine. They probably have some type of an association where it's mutually beneficial. This thing here, I figured out what this thing had on it by accident. We were late at night, we were dead tired, we got our specimens, the vehicle came up, we're in the lab, and I forgot to put on my latex glove, and I picked that thing up. And probably for about 12 hours, my hand stung from all the hydroids that were in it. Though I won't do that again. This is all those little spikes are stinging hydroids. So nobody's going to want to eat that. And now I've got a couple of natural sponges. They aren't glass sponges here. But if anyone guesses what these hairy things are on this sponge, I'm going to throw a sponge for you. This is a hard one because I did not know what this was either. And I'll tell you, it's not a hydroid. Okay? All right, I don't hear it either because I didn't know as well. This is a black coral. And it's specialized to live with these sponges. And when most of the time when you see these particular sponges, which is in a genus called Phorea, they've got these black corals growing with it in a close association. And they just described this a number of years ago, that black coral. And so it's totally unique type of associations that they're developing for some reason. Okay, but aside from animals that have really close associations, a lot of animals use sponges to get up off of the bottom. They'll perch on them because they get them high. Like these little muted crabs right here. These are feather stars. They're in the group, the crinoids, snails. And here's another one. If anyone knows what these pink and red animals are, they get another one of these sponges. Any guesses? A hint, it's got two things coming off of it that are retracted. What's that? Who said that? That's a ctenophore. Those are the things that float around and light up and they look all pretty, but down in the deep sea, they reverse things. The adults are what is typically the larval form of the floating ones, and then the juveniles are the floating pelagic ctenophores. It's a benthic ctenophore. Those are the teeny. They're normally stringing out from the side of this thing, and they love sponges. So anyway, has anyone heard of the Venus flower basket story? The love story, the deep sea love story. Has anyone heard of that? Got a couple people. Okay, I was looking at this, and Japan is very famous for this kind of thing. And I was looking at, is this true? And they were commenting saying, well, Japan supposedly does this maybe in one prefecture, but they're not really sure this is really true. But anyway, there's a couple of shrimp called spongicola. You can see one right there, and I think one down there. And they enter this sponge when they're larvae, and they get inside, and then they start growing to the point where they can't get out because they're too big. And they are trapped for life inside that sponge. And they wind up having one male and one female, and they live together for their entire lives inside the sponge. They can reproduce, and all of their product goes out because it's small out through the top, and they go find their own sponges, right? But it's a deep sea love story, and supposedly in Japan, they were giving these sponges as wedding gifts because it symbolizes love until we die type of a thing. So kind of a neat little story. Okay, up here in British Columbia and up in Alaska, you can get these amazing sponge reefs everywhere in shallower water because it's dark and cold. It's very important ecologically, not because the animals perching on it, but there's so many animals that are dependent on these reefs. And here's something down in the Antarctica. You can have sort of this big, huge reef, and also you can have very dense communities of these sponges. This is something that's called an Hexactinellida jubini. And they estimate that one of the members of the sponge, I'm not sure it's one of these, but one of these sponges is estimated to be 15,000 years old, that particular individual. And it's based on, I think, in part, they found the sponge 10 years apart, and they measured it again, and they could find 0 growth rate on the thing, but it's perfectly healthy. So it's an estimate which would make it one of the oldest animals in the world. But then this guy came along. Hang on. Here we go.
This looks like the same folded sponge that we sampled yesterday. Look at the size of these things. Oh my goodness, where did this guy come from? Holy cow.
That is unbelievable.
Oh my jeez.
Absolutely beautiful.
Bruce pointed out he's not sure which is bigger, the ROV or the sponge. And looking at the Ceros view overhead, I'm not sure. I think it's a tie here.
This is the largest thing I've ever seen underwater.
People may be wondering right now if they're looking at this, how old is this thing?
And sponges are extremely difficult to age.
Thanks to the work by Les Watling, Papahanaumokuakea is home to the largest coral in the world, and now perhaps it might be home to the largest deep-sea sponge in the world.
Okay. So Daniel, the other voice you heard on that clip, we were the two scientists out on this cruise up in the Papahanaumokuakea Marine National Monument in the northwestern Hawaiian Islands. And he said, you know, we ought to write this up. So we went and we got on the ROV with measuring tapes, got the pilots to let us measure everything on this thing, measured it all up, and then looked up to find out what the largest sponge was on record. And we wrote up a little 2-pager. And with a little question mark, biggest sponge in the world. And the reason we wrote it up is because I couldn't even figure out what genus the sponge was. I had never seen this before. And it was more like an alert that we've got some really big animals down here that are being discovered. Okay, well, what happened after this little 2-pager? 14,000 news stories came out of this thing. This is one of them. Everywhere in the world, radio, TV shows, Papua New Guinea, Czechoslovakia, Radio Australia, Canada, 14,000 news stories, supposedly one of the more influential publications of 2016. And all I can say is that had to be the mother of all slow news weeks. It's a sponge. Anyway, NOAA, which we were out on their boat, they were pretty happy about that. Something like $25 million worth of internet ad revenue came out as a result of that. But anyway, okay, so enough of that. Now we're going to go to skeletons here. So this is glass skeletons, and this is what is a really cool topic. I'm going to start with a couple drawings here. But basically, here are your 4 groups, classes in the porifera. And this is calcarea, and the reason it has this name is because it makes its spicules out of calcium carbonate. It does not make them out of glass, or silica, if you will. These other ones, they've got a few that make them out of silica, but this one, homoscleromorpha, is referring to the fact that its particular spicules, the sclerae, if you will, that's what that part is, are pretty much homogeneous. They're tetraxons or 4-rayed type of things in their body form. And then here, the demo sponges, that actually translate to common sponge because this has got over 6,000 species in it. They can do a variety of these triactons, tetractons, and so forth. But the fact of it is, the Hexactinellids are the only group of sponges that use a 6-rayed spicule as the base. Now, that doesn't mean that every single one of their skeletal parts has 6 rays. They can be reduced or lost, but they are derived. The ones that don't have 6 rays were derived from an original 6-rayed. And it is the only one of the group, and that's why it's named Hexactinellida 4-6. That's what I'm going to talk about for the rest of this thing, the Hexactinellids. And a couple things I want you to know about. They are the only animal that makes their skeletal parts out of glass. You have some some small unicellular animals and some plants that use silica or silicon dioxide to make skeletons, but these glass sponges are the only ones to do it in the animal kingdom. You don't have to read through this, but one of the reasons why they're talking about possibly deep-sea sponges using glass is because there are things that are called these horizons. For example, there's a aragonite saturation horizon, which refers to calcium carbonate. And it means that calcium carbonate is at saturation above that horizon, and once you get below that horizon, you start getting out of saturation, which means it's harder for animals to actually acquire calcium carbonate from the water to use in shells and things like that. Well, it's kind of the reverse situation for deepwater sponges, where there's sort of a supposed variable silica saturation horizon, where below it, silica is saturated because of all the diatoms and everything that drops to the seafloor and are slowly going into dissolution, whereas in shallow water, the silica is lower. Anyway, I don't know why that's a fun fact, but I guess it is. But anyway, that may explain why the glass sponges are down in deep water only for the most part. How do we make glass? Well, this is what we do. We get some sand that's a silicon dioxide sand, and we add soda and limestone, and we can put colorants and everything else, and then we heat it to 1,500 degrees centigrade like that. And that's our technology here, how we make glass bottles and things. Sponges do things differently. Okay, now you're seeing the largest spicule in the world. This thing is 9 feet long. It was described by Wang in 2011, his group here, and it's not the largest sponge in the world, even though it's got the largest spicule or skeletal part, because there's a sponge. It's a little tiny thing on the top of this big, long spicule here. But what that enabled people to do is to look closely at the structure of these spicules and try to understand how their sponge is making them. And what they found out is they have this core area called the atrial filament or atrial canal in here, and it's got a proteinaceous type of center area here. And then from that canal, they're creating layers, thin layers of silica on top of this, or glass on top of that, going a little bit further out and out and out, and that shows you sort of an electron micrograph of that whole process. And so that gives us some clues as to how spicules are being made in the first place. That's a little bit in dispute right now. And then there's another paper that came out in 2017, and this is not on a Hexactinellida, it's on a demosponge that produces glass spicules. But it's a really cool paper because what they did is they looked at a little bit more complex type of spicules. So here's your single ones here, and they used 3 different species of these siliceous demosponges. And what they found out is that each of these produced sort of a proteinaceous core, like this, and that particular filament could be branched or it could be single, and then, or it could be this very complex sort of spherical type of protein network. And then they lay silica down, silicon dioxide, and they convert, I think, something or other acid, a hydrated form of it, into silica, and they lay it down onto that proteinaceous scaffolding until they finally get the final spicule. Okay, this is an amazing process. The reason being is because it's done in freezing cold temperatures, unlike the way we make glass. Freezing cold temperatures, they use a template or a scaffolding, and these are some of the results. These things are about 25 microns in diameter, because when you're using protein filaments, you can get to all types of detail in how you make these things, and it is absolutely marvelous. In fact, these guys, if you read that top quote, basically saying that sponges have figured out a way to actually make glass that is so far beyond what we're able to do ourselves. And they're 500 million years old, right? So this is a process that was started 500 million years ago, and now we're just kind of discovering the general concept of having some type of a scaffolding or a template, which then we can layer very thin layers of products on there to make electronics and all types of things now. But they've been doing it for 500 million years, and we still can't do it with glass. But they can, and that's just remarkable. I'm going to show you some more examples of these, but what are they doing with these things? I don't have a clue. Why do they make all of these various types of structures? Well, a lot of people have been thinking about that for many years. This is, has any of you heard of the camera lucida technique? Some of you? Okay, great. This is sort of an example of an older camera lucida, and what that means is that back in the mid-1800s, when sponges were first being investigated and the spicules were being used, the glass parts were being used for their taxonomy, they could look at a microscope, but then how do they translate what they're seeing? Well, somebody came up with this idea where you have some type of a mirror type of arrangement on these things that projects what the microscope is seeing onto a sheet of paper, and the researcher can then just draw or trace the microscope image on the sheet of paper. And that's what you can get. That's all of these were drawn using that technique, and it's still being used today, the camera lucida, because it is an effective way of actually depicting the spicules. Okay, well then, of course, we got microscopes with cameras on them, but here's the problem. You want to get a lot of detail from these glass structures, but when you zoom in at 40 magnification or higher, you're getting a very narrow plane of focus on it, and this is a 3D structure, right? And so this shows you what happens. I mean, you get only a little bit of it in focus at a time, and so this is actually crappier image than drawing it on paper, trying to use a microscope camera picture. So then in 1965 is when the first scanning electron microscopes came out commercially. They were invented earlier than that, and that solved a lot of the problems. Okay, scanning EM is basically totally cleaning the spicules, then applying an ultra-thin layer of gold onto the spicules and then bombarding it with electrons. And you can get just absolutely fantastic images. So this is 50 microns, or 50 millionths of a meter. And that's 5 millionths of a meter. So this is one of these little arms that they're showing in detail. And then this is the top part right here on it as well. And this is a way that, this is the gold standard. It's just gorgeous what they can do with an electron micrograph. And so what I want to get onto here is spicules have been classified in two different size categories for many, many years. Megasclerae, which are ones that you can actually see with your own eye. That doesn't mean that they're like a stick or anything like that, but if you look in a little dish, you can see something down in the dish. You can actually see that there's something there. That's a megasclerae. And then and these are some examples of these right here. They tend to have more rays on them as well. This is 6, you can see all the 6 rays. That one lost the top one, that one lost the top one. There's the little nub. I don't know if you can see this one. This is called a diactin, or a 2-rayed spicule. And if you look right there, right in the center, close up, you can see 4 little knobs around it. Those are the other 4 rays that are reduced on that thing, proving that it's actually a 6-rayed spicule that it was derived from originally. And these are one of the anchorate ones as well. But these things tend to be, it can be a couple millimeters in size down to maybe half a millimeter in size. The thing about these megasclerae is that they can actually glue them together. So some species will keep them loose in their tissue, and these are softer kind of glass sponges. Others, they'll actually glue the spicules together to form a rigid structure. And this is an example of one, and the way you look at this is there's the center of it right there. There's 1, 2, 3, 4, and there's a ray coming out here and one coming out the backside. And then these 2 are right next to the next one. The 2 rays glue together here, and it goes to the other nucleus, or I don't know which, I've forgotten the term for the center of the thing. But anyway, so you can create these beautiful little square things by gluing your spicules together with added silica. Okay, and this is really, really important for the taxonomy because they can glue them together in all different types of ways. Some of them are very, very uniform like this. This is what's called a ferrioid skeleton. And then you can get all these sort of ones that are kind of glued together hopscotch all over the place. You know, this tip will glue over on this part of it, and this will glue over here, and it creates all this sort of mess. But it isn't a mess because you can see that the skeletons can actually be created in a way to form very clear channels, which are very, very important for sponge flow and everything else. Some of them put a lot more silica on them to make them very durable, almost rock-like solid as well. So these are called dictyonal frameworks, and they're very important in diagnosing the sponge taxonomy. Now, these are the microsclerae, and these are ones you cannot see with the naked eye. They're too tiny. The only way you can see them is with your microscopes as well. And again, I don't know why they are making these things. Some people talk about anti-predator, but 25 microns, whatever eats, it's going to go right through the system. I mean, how is something like that going to bother some predator eating that? It's round, it's just going to go right through them, no problem whatsoever. But look at all the shapes. This is called an amphidus. These are called discohexasters. These are aspidoplumicombs or something. I've forgotten what the name of them. These are anchorate hexasters. And these are these look like 6-rayed standard ones, but myriad of different forms and shapes, the function of which we don't really understand. Okay, I and a couple other people think there ought to be a 3rd class, and that's mesosclerae, because there's a group of spicules that's right in between the big ones and the smaller ones, and they don't really get annealed to the frameworks very much. So I'm just calling these mesosclerae as well. And these are some examples of some of these. This is what's called a scopule. This is called a clavule. And the 2 of these together are called septules. And that name is derived from a king's scepter, holding up his staff with the top thing on here. There's a spiny pentactin. These are called pinnular hexactins as well. But again, the detail and how they're making these things and why. These you can possibly imagine that these could have some type of a defensive type of a reason. And some sponges will actually stick these out their sides, so they probably are defensive, anti-predator. But for these other ones, who knows? Who knows why? It's structural, it helps with the integrity of the sponge. Okay, the thing about scanning EM, scanning electron microscopes that you just saw, is that it's expensive, and you have to get your sample ultra clean, which means you're usually bathing your spicules in fuming nitric acid. Okay, and it takes a long time, and to actually get good at this takes a long time. So the actual progress of naming all of these undescribed species of sponges is very, very slow. So one of the things I'm trying to do is to see if could be some acceptance for doing it a little bit different. It's not the gold standard of getting spicule imagery, but it's a little bit below that, and that's taking a standard microscope with a camera on it, piping it into a computer that has EDF technology on there. There's software in there. I don't know if you folks are familiar with the concept of extended depth of focus, but what that means is I can take a picture here at the top and then take a continuous set of pictures working my way down. And that may seem tedious, but when you get going on it, it's like click, 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 and you go right through the whole spicule, and then you press a button, and the software puts it all together into a single in-focus object right in here. And so these are various examples of that that I've taken pictures. This is my little setup at home. And you can sometimes, I've forgotten what Diane said about my home lab. And there was actually a news article recently about some home lab that was getting busted. I don't think I actually have to worry about this because my chemicals are, let's see, distilled water, Clorox from Safeway, and denatured ethanol that I have to order. That's just to preserve the sponges and the spicules themselves. And those are my chemicals. So I think I'm probably okay. I'm not going to get somebody busting me for my home lab. But here, because you're using Clorox, you don't always dissolve all the tissue 100%. So you can go into Adobe Photoshop and just clean that up a little bit so you can get nice clean spicules. So they're not scanning EM, they're not gold standard, but they're pretty good, and they could be used to describe some species here. And so I'm going to finish up with just description of species a little bit and show where that's going right here. This is Townsend 1913, and this just shows you that back then in 1913, they didn't have remote vehicles, they didn't have submersibles, they didn't have this. How are they getting these deep water sponges? They were trawling them, they were dredging them up. And the sponges would come up in pieces. So you get this type of stuff. And they would photograph these pieces. They'd do their camera lucida drawings on the spicules from these pieces. And they would describe some of the channels in there, the ones that have the dictyonal frameworks. But that's it. You don't really know what that sponge looked like on the bottom because it couldn't get the whole colony. Okay, nowadays, this is 2011. These are some of the sponges off of New Zealand that were done by Michelle Kelley and Ricewig, and beautiful, beautiful EM. But the problem is New Zealand didn't have an ROV and didn't have a submersible, so they were dredging all of theirs up. And here's a fragment that shows what they were able to get the spicules from it. You can identify what that family is in that genus as well from all of this. But you don't really know what the whole sponge looked like, and that's kind of a problem. Because you can't do any ecology unless you know what the colony is. You can't collect everything you see down there. If you're going to do a transect and try to see what the density of this particular type of sponge is out there, you got to be able to ID it right on the spot from video. But if all of the taxonomy is based on spicules and not their colonies, then you got a problem. Okay, so this is a paper in Shen from 2022 describing a species in a subfamily called Bolosomini. And anyway, if all they presented was the lab specimen, like the other thing, that's what you would see, and you'd see this beautiful scanning EM of the spicules, but they collected it with an ROV. Okay, and so in this case, you got the colony shot. And guess what? It had a stock, which we wouldn't have known about unless we actually got the in situ image. So you can see how important that is because that's going to be part of the diagnostics for this one. Here is one that I'm going to try to submit with a Russian colleague right now who's in Moscow working at the Shirshov Institute. And this will be the first time I've actually gotten the courage up to try to submit this for review on this. This is going to be a new species in a genus called Periphragella. And these are all my pictures that I took from it as well. This is what came up from the ROV. Well, duh. What are you going to do with that, right? But, of course, it was collected ROV, so we now know that the colony looked like this, even with lasers on it to give an idea of the size. And that's how the taxonomic descriptions are headed. Now, everybody who's putting in new species now are adding the colony shots, even lab shots, spicule shots, and in much more complete package. But there's one final thing too here, and this came out in 2017, and I was fortunate to be a co-author on this paper. This is a German geneticist, Martin Dorman, and he was one of the few people who knew how to do genetics on sponges because they're notoriously hard to do genetic work on. And basically, he got all these people together to get, I was responsible for all the morphology in the colony, in situ colony shots, and they got Michelle Kelley and Henry Reiswig to do the scanning EM and then his genetics, and he put them all together, and he completely revised 2 orders of the Hexactinellid, combining them into an order Septulophora, because he was able to get all of this information and pile it all together. Now, to actually change taxonomy on the order level is extremely rare in this day and age. It's almost impossible, okay? But this is not something about how great our technique was getting everybody together. This is something about how little we know about sponges. Yeah, we just, with this one paper, we realized we don't even have the orders right on these things. We still got to get all that straightened out. There are hundreds, if not thousands, of undescribed species of sponges still down there. And a lot of them are in museums already that just haven't gotten processed and analyzed as well. So it's a big task, and they're a wonderful group of animals to work on. And I want to thank you all very much. If you're interested in deep sea stuff, here's a couple of sites that you can go to and see sponges as well as other animals. And appreciate you coming.
Thank you for a great talk. The laying down of glass layers upon the protein core, is that done by a live layer similar to a xylem and phloem in a tree trunk?
It's my understanding, and I don't understand it all that well, and it's also being revised right now, how it's done, is it's an enzymatic process. In hexactinellids, they actually have 4 proteins. Hexactin or something like that is one of 4, and they have an actin, and somehow these proteins are working together to create the scaffolding and actually get the hydrolyzed silica to actually get onto the scaffolding, but it's way beyond me.
So it's from the inside, not the outside, not a live membrane on the outside.
Oh, that's a very good question. I think it's from the inside, but there could be also some thin layers of some of these other proteins that are facilitating on the outside. Yeah. Yes and yes, it's a small group. And yes, it's probably a dying art, at least the traditional taxonomy. There haven't been a whole lot of people getting into the genetics because it's so hard to come up with the right markers, and there's so little tissue on them. But there's a group in China that's recently put out some really good descriptions. So that's a new group that wasn't around before. This is not the Wang who did the giant skeleton. It's the Shen et al. And a couple of his colleagues. They're doing really nice stuff. So China is picking up the ball a bit here. In Russia, there's Konstantin Tabachnik, who I work with, and he's been doing this for decades. Henry Reiswig used to be up at the University of Victoria, and he lived up north of Victoria, and he passed away, very unfortunately, a number of years ago, and he was like the world's authority. As far as new people, well, Martin Dorman in Germany is one. Michelle Kelley down in New Zealand is another, but they're all spattered all over around the globe, and I don't know a whole lot of new graduate students who are doing a lot of this type of thing.
And then my other question was, who does the information get submitted to you? When you kept saying they, you submit it to them. Is there a board, a scientific board that does the order reclassification, or what does that look like? Who controls that?
Okay. Well, when you're going to publish a new description of the sponge, you're going to pick a journal, and then you're going to submit your manuscript to that journal. And that journal will then pass that manuscript out to probably 3 reviewers that are all knowledgeable of the sponge world to evaluate whether it's worthy of being published as well. And if they decide it is, then it gets published. Then it'll wind up getting, if it's a new name, it'll get into what's called the World Porifera Database, which is an online resource. It's a gigantic catalog. And there's also another database that's associated with called WORMS. It's the World something repository of marine species or something like that. I've forgotten. And the new names will get incorporated into these international databases as well. And those are the main things. Now for ecological work, when we're actually doing surveys using ROVs or submersibles and counting and identifying animals from just seeing the colonies, that is now going into the Deep Sea Coral Research and Technology Program's online database, where they have hundreds of thousands, if not a million, records of deepwater corals and sponges there, so you can see what has been recorded where in the world. And it's a US NOAA type of a thing, but and unfortunately they got severe funding recently, so they're down to like one person maintaining that database. But anyway, they accept records from all around the world.
Chris, in the fossil record, say the early Paleozoic, spicules are not uncommon, but seldom the rest of the creature. Would that be because these glassy structures just don't preserve well or break up under subsequent sedimentation, or why would that be?
The spicules are, a lot of them are preserved in the fossil record, but a lot of times, if it did not create an actual framework, a dictyonal framework, back then the spicules, when the actual tissue decomposes, they all scatter everywhere, and then they're hard to see in the rock because they're so tiny in there. And so that's part of it. When there's some larger spicules for certain types of sponges are left on, or fossilized, then you can see those, like that example I was sort of showing you. But that could be it. The glass usually persists for quite a long time, the actual silica. But again, it's like anything else. It may dissolve more slowly than, say, calcium carbonate, but it is going to dissolve over millions of years unless it gets buried and somehow preserved.
Very interesting talk. Nice to meet somebody else who's worked on sponges.
Yeah, we got to stick together.
Yeah, I know. If you spend enough time on scuba and you know what to look for the demo sponges at least, you can see when they grow up next to each other on the substrate and fight for space, they will either kill each other at the interface or they'll put up a wall. And so all of the time that you've been observing these deep water glass sponges, have you ever seen anything like that for glass sponges? I don't think there's anything in the literature at all about whether they defend themselves for space on which to grow.
I have seen that giant sponge, the biggest sponge in the world, it actually had like 2 or 3 different species that were growing on it, that landed on it. So the example I showed of the freeloaders getting up on the dead sponge, that's one thing, but they'll get up on live sponges, and it doesn't seem to cause any type of reaction between the 2 sponges. He's just hanging on and grabbing onto this sponge, and I've seen sponge colonies that are butting up against each other, but I never saw any type of a wall or never saw any type of a toxic interface happening or something like that. So I guess it's quite different.
Well, the wall might be microscopic, but it's interesting that they don't, I mean, clearly they're not fusing together when they're different individuals. Okay, good.
I have so many questions. This was a great but I'm only going to ask one. It seems to me that if I could transport myself back 200 years or 300 years in the sort of history of zoology and natural history and the like. This is about the stage where so many of those efforts might have been. And I'm curious, more than curious, about the ecology. And I know you can't really tell from what you've got so far, but I'm wondering if you have any speculations about the deep sea ecology that allows these things to flourish, and what else is going on, and what sort of relationships might be going on in that world.
Okay. So once you get down below about 1,000 meters or so, the two large, most dominant animal forms that you see down there on rocky substrates are going to be deep sea corals and deep sea sponges, filter feeders. The deep sea is populated by a lot of filter feeders. And so these sponges are there as well. Sponges seem to have very adequate defenses against predation, so they tend to thrive, even though they're extremely slow growing, we think. They tend to thrive down there because no one's bothering them for the most part. There's a sea star that supposedly eats sponges. I've seen these sea stars on sponges, but when you see a sea star on a coral, the sea star cleans off all of the tissue, leaving a bare skeleton of that coral. But in sponges, you don't see that, as though it's just kind of scraping along, getting a little bit of tissue, but it's not really bugging the sponge at all. It's just nibbling on it, something. So they're not really getting predated upon. They don't have a ton of competition, if you will. And the sponges, one of the things I didn't really get into, but with this type of cell called a choanocyte, they actually can create their own current because the cells are flagellated and they're constantly beating. And they can actually populate areas where there's very, very low current, where corals cannot, because they can make their own current and get water passing through their bodies, whereas the corals got to have some type of current flow in order to feed on there. So they're very important. A lot of animals use them for protection. They go into all the holes and cavities, and that's where they live. So crabs and other types of things, they find shelter in these. They're very important for small animals, crabs, brittle stars, things like that, that would get predated by deep sea fish. They can filter enormous volumes of water, and so they're taking a lot of stuff out of the water column itself. Now, what effect that has on the environment, I'm not really sure. But I don't know what else I really know about their ecology, except they're very common. They're not everywhere. In some places, you'll see them scattered all around. In other places, you'll find this immense community in which we're trying to understand, why is this place so rich and this place not so good? So, too many questions.
Yeah, really.
Yeah, that's why it's fascinating. We don't know yet.
Hi. So I know deepwater fish die from decompression when you capture them, but do sponges suffer the same consequences when you bring them up from the deep water?
Nope, because not all deepwater fish suffer from decompression. The ones that have air bladders do, because the air will expand, or gas bladders, if you will. But then a lot of deep sea fish have a lipid-filled gas bladder, so they can move fairly extensively in the water column. But the other problem is that if they're really deep water, below, say, 1,000 meters or so, there's things that are called pressure-sensitive enzymes. And if you bring up a fish that has a pressure-sensitive enzyme, it's going to die because that enzyme just denatures completely and becomes non-functional as well. That could happen with sponges. But I think part of the problem is that I don't think anyone's managed to bring up these glass sponges and keep them alive. I'm not sure if they've even tried to do that. And if it were possible to do that, then maybe we could start to understand their physiology a little bit more.
Cool, thank you.
Well, thank you all so much for being here. And Chris, thank you so much for your excellent presentation today. And
thank you. I appreciate it. Thank you for coming.